Next speaker is Yu-Xuan Lu, and he is from the Max Planck Institute in Cologne. Hi, can you hear us? So whenever you're ready, you can just share your presentation and start.
Okay, so thanks a lot to inviting me to give us such a fantastic conference, especially thanks for Morten, Alex, and Daliana for organizing this meeting. Thanks a lot. So let's just get started. So today I'm very glad to present one of our fantastic projects, basically doing the drug repurposing to find a new target for the old drugs. So I'm Yu-Xuan, as Diana already mentioned, and from Max Planck Institute for Biology of Aging in Cologne, so basically. And first of all, I would like to give a big thanks to my boss, Linda Partridge, but also all the collaborators, which is heavily contribute to this project. And as many of us, no matter whether you are in Copenhagen or around the globe, when you attend this meeting, I assume that many of us know the importance of intervention that can slow down aging and the onset of age-related diseases. For instance, like genetics, drugs, metabolite, and diet, and of course also exercise. And many of these intervention and challenges we already know that can influence the hallmarks of aging and eventually to lead to improve or slow down aging and aging-related diseases. And today I would like to really want to give you one of my topics about the drugs. So what exactly drug, or we call it pharmaceutical interventions? And so far, we actually know there are several drugs, including rapamycin, metformin, and spermidine, and also a lot of good things. So some of our speakers already mentioned previously that can attenuate aging hallmarks, and some of them or even show experimentally can extend the lifespan, at least in some of the model organisms. But when you look into this chart, you also are actually still find that there's a lot of gaps that need to be fulfilled, even, for instance, the rapamycin, what we are going to talk about. So why is that? Because if we look into rapamycin, for instance, we know that actually a couple of decades ago that rapamycin can specifically inhibit mTOR C1. So basically it binds with, if go with FKP12 and then bind to the mTOR C1 complex. But this, even though this is a very specific target, it can influence a lot of downstream signal, for instance, the phospho S6 kinase, 4EBP, and also autophagy. That is actually to an issue that when we want to imply this for cure aging, because a lot of this drug, when it developed, is mainly just for acute improvement, for instance, like change the cancer or the tissue transplant. But when we work on aging, we need to consider there's a lot of effects we don't know at the moment. And if we just immediately use this drug for human, for instance, at least for my opinion, is very risky because we just doing something more or less a centuries ago that just, selling the Titanic and going to hit the iceberg, actually we don't see. So what we are doing is trying to step one step back to trying to figure out if a rapamycin, for instance, have something else effect, at least on the organism, and see what we can actually trying to deal with that and improve our understanding for these drugs. So we are actually glad to trying to use the Drosophila, the fruit fly. The reason for that is that there are a lot of high similarity between the human, the mouse, and of course a fruit fly. So as you can see here, couple centuries ago, Da Vinci already wrote a couple of paintings that's to indicate that fruit flies are more or less similar to human. So obviously they have organs, they have sex, and they also can eat often. And so that's the reason we start with fruit flies. So first of all, we use a rapamycin treatment, which we already know that, and also a lot of labs show that it can increase lifespan in female flies. So as you can see, the figure A, it more or less increase 10% lifespan, extend 10%. And we're trying to do it a systematic way, to collect the fly from young, mid-age, and old time point to dissect them into 4 main tissues. So brain, muscle, and fat, and intestine. And then trying to do the Western blot to test what kind of a protein that it will be influenced by rapamycin. So very surprisingly, actually, we found that rapamycin treatment can increase histone proteins level in gut only. So as you can see in the figure B, that when you treat when the fly eat rapamycin and we dissect them, we saw that at least the histone H3 and H4 are dramatically increased response to rapamycin after 10 days. And when we also do another Western blot, we even show that after only 2 days, the rapamycin can already significantly increase the histone protein levels compared to control. But that's only happened in the gut. But what exactly is doing of this histone? So we're trying to, go into use the immunofluorescence to try to figure out if a histone will influence, for instance, the chromatin, because we know that the histone is one of the key components, to organize the chromatin. So if you can see in the figure A, you would appreciate the gigantic change of the nucleus. So when there's no rapamycin, actually the nucleus, as you can imagine, just like a round, like a ball. But when you treat it with rapamycin, you see the chromatin, or like a nucleus, become a donut or a black hole, as you would appreciate. And that is something we can actually mimic by expressing histone H3 and 4 together only in the gut enterocyte. So that means the amount of a histone, at least the histone 3 and 4, and the gut enterocyte can directly influence and chromatin organizations. But what exactly does? Of course, we all want to know if this have some input or some influence to aging. So we check the intestinal dysplasia, which you can see the figure A, and in the normal situations, when the guts go older, we will have develop some pathology, for instance, like the gut tumor, the monolayer stuff, the multiple layer stuff, as you can see here, for the yellow arrow. And when we treat the rapamycin, usually can inhibit the gut pathology. So basically have the rapamycin treated gut have less gut tumor compared to control. And when we overexpress the histone H3 and 4 in the gut enterocyte, it mimics the rapamycin effect. So, which means that it also can improve the gut, inhibit the gut pathology. And more importantly, if we look into figure B and overexpress the histone H3 and 4 in the gut enterocyte, can extend the lifespan to the same degree as the rapamycin does. And when we combine these two interventions, and it will not further extend the lifespan, that means histone H3 and 4 and rapamycin go into the same pathway that suggests. And then we're going to go back into a bit to trying to understand what exactly histones increase histone H3 and 4 does at a molecular level. So obviously, one of the things we're trying to think about that is it might change the histone modification and also influence some gene expression. And so first we did the iron-seq, and we found out that there are a couple of autophagy-related genes are upregulated under the overexpressing histone H3 and 4 in the gut enterocyte. As you can see here, the Bchs, DOR, and STAT92E, something like that. And we're trying to use the qPCR to verify them. And Bchs and DOR, which is something response to overexpressed or histone H3 and 4. And when you look into the figure B, we also use the chromatin immunoprecipitation, so the CHIP, to find out and at least several of the histone modification, including the K4 trimethylation, K9 trimethylation, that's K27 trimethylation, that's somehow change when you manipulate or actually increase the histone H3 and 4, which is in line with gene expression we did. And so as So we found this autophagy-related gene might be important for the histone-induced lifespan extension and health span. We pick up one of the well-known gene, which is called Bchs, as you can see here, and we trying to either knock down the Bchs, which you can see in figure A, which that's block the increased histone H3 and 4 effect on gut health. And it also blocked the lifespan extension that histone H3 and 4 helps. So basically, it can tell us that the Bchs is required for the histone induced gut health and lifespan extensions. But what is actually important, because we know that for those of the staff, we always want to know more about, whether this is applicable. So we also took the mice, and as you can see here, the Rapamycin treatment also increased the histone in mouse small intestine, similar to the gut, similar to flies. And it also changed the chromatin organization and also the gene expressions, which is called WDFY. So the Bchs homolog in mice small intestines. So for the summary one, we are very glad to show you that beyond the classic TORC1 cascade, so we also found that actually there's unconventional and intestine-specific TORC1 histone exits that go through rapamycin, histone, and Bchs, which is increase the autophagy and at the end of the day, improve the gut health and also slow down the autophagies. But that is not the whole story, because when we talk about, actually, we only focus on the females. And we might know that what about males and men, because that is also very important. So we actually, when we look back, we know that actually many aging interventions show sexual dimorphic response, including rapamycin, as you can see here. And rapamycin actually have a bigger impact on female than males in terms of lifespan, at least in mice and also in flies. And there's a couple of possible mechanisms explaining female why response stronger to rapamycin to males. But since there's a very complicated situation, at least that we would like to use a fly to detangle these interactions. So first of all, we're trying to treat the rapamycin, use the rapamycin treat the flies, and we found that female responds to rapamycin treatment, but not males. So as you can see here, the blue line, there's nothing happened. And but interestingly, when we test the phospho-S6 kinase level, it responds to rapamycin in both sexes. So basically, the drugs can reach to the target tissue, have the same level to, the female and males. But why, interestingly, on the other hand, it only increased autophagies in female, but not in males, because the male is already very high and rapamycin cannot improve even further. And so we want to say whether the autophagy is very important and to, have this sex dimorphism response. So we're trying to suppressing the autophagy in intestinal enterocyte by just knocking down the ATG5 in the enterocytes. So as you can see here, when you knock down the ATG5 in the enterocyte, it is shortening the male lifespan, but have nothing to do on the female lifespan. That means autophagy is important for the lifespan in males. And as I already mentioned before, we want to check whether histone have something connection to the response to rapamycin. And we found that actually the level of the histones and Bchs are increased in response to rapamycin in female, that's only, but not in males. So now we know there's a sex difference, but can we actually do something actually? And I would like to just say 2 or 3 more slides to talk about this. And as you might know, that actually sex determination pathway in Drosophila is very relatively simple, because it comes from the sexolethal and transformer and then the double sex, so which is determine the female or male morphology on behavior. And you can actually do switching sex only in specific tissue. For instance, here I just switching sex in gut enterocyte by overexpressing the transformer or knocking down the transformer here. And we found that actually when we switch sex, for instance, we feminized the male gut, we found that the histone, Bchs, and autophagy level actually in response rapamycin again after we switched the feminized male gut. And surprisingly, actually, these feminized males and also start to respond to rapamycin, and it can extend the lifespan again. So that is really cool because we can actually try to use this technique maybe for the futures. That will also benefit for the human, hopefully. And so today I would like to just thanks a lot, and I gave you the two stories. One is the new target, and which means that the chromatin, the histone can, the rapamycin will directly influence the histone, which is influence the histone modification. At the end of the day, have something to change the epigenetic and autophagies. But this will also interact with the cells. So that's at the end of the day, one sentence can wrap up this talk is we found that unconventional intestine specific, maybe also sex specific TORC1 histone axis that will maybe do with something we will have hopefully a new target for the new pharmaceutical intervention for aging. And if you want to know more, we have actually the last chapter of this trilogy, lifespan extension drugs, the rapamycin. My boss, Linda Bchs, who will be the last speaker at the end of this talk and this whole talk, and she will speak about that. So thanks a lot. I'm glad to take your questions now. Thank you.
Thank you very much. We are running a little bit short in time, so I would recommend you to go on Slack and to answer the questions there.